Can we synthesize such a type of a animal or the plants which are having its one of the gene derived from the human being? Can we synthesize the plants which can glow at the night time? Now it is a very possible because of the techniques which have evolved in the genetic engineering. Hello, this is DM Vactor, working as assistant professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. And in this video, we will discuss about the cloning vectors. We will also focus on one of the cloning vector, that is plasmids. Before starting, we must clear that what is a cloning. If we identify the term cloning, it will indicate that it is the production of population of genetically identical cells or of organisms asexually produced by a single cell or the organisms. Whenever there is a group of cells whose genetic material is identical, which have synthesized by the method of a reproduction and its genetic material is identical, at that time that group I will refer it as a clone. And now this is another terminology for the clone, but here we are using the cloning vectors for the cloning of our DNA. That is, we want to synthesize or we want to produce our gene of interest in the multiple of copies, not using PCR, but here we are using one short vehicle which is made up of a DNA and it is called as a cloning vector. Here we are cloning our DNA and not we are preparing any such a clone or identical animals. Now, how you will define this cloning vector? This cloning vector is nothing but the small piece of a DNA which when enter into the bacterial cell or any host cell, then it should have the stable maintenance. That is, it will not react or it will not depend upon the host replication machinery. It will replicate itself. It should be stably maintained whatever organism or in which cell it has entered. And so the cloning vector can be derived from the virus, it can be derived from the plasmid of the bacteria, and nowadays genetic engineers have synthesized such vectors by artificial way. And due to these cloning vectors, it is now very easy to transfer any gene from any organism into the specific host cell. Now, how it takes place at genetic engineering level? Suppose this is a cloning vector and I am having my foreign DNA and I want to transfer this foreign DNA into the specific host cell. Now what I will do, I will use this small vehicle to transfer my gene into the specific host cell. So I will take this foreign DNA and I will ligate this DNA into this cloning vector in such a site that this site will be not so useful for this entire structure. So the not useful structure I will remove and then I will ligate my foreign DNA to this vector. Now this vector will be termed as a recombinant vector because I have recombined my gene of interest with this cloning vector. For example, suppose I want to prepare human insulin, then I will isolate the gene of insulin from the pancreatic cell. Once I am having insulin gene with me, I want to multiply that copies and I want to produce that insulin into the specific host cell in the mass production so that I can sell that insulin at market. Now for such a artificial synthesis, I will take one of the vector which is a plasmid. Now the plasmids are present in the bacterial cell and from that bacterial cell I will isolate this plasmid. I must know the entire map of this plasmid. After that only I can cut the unwanted region on the plasmid and then I will paste my gene of insulin with that plasmid. Now this plasmid is nothing but the recombinant plasmid because we have recombined our gene of interest for this vector. Now this recombinant vector I will transform into another specific host cell with the help of different methods are there I can use the calcium chloride method. I can use the electroporation method, by using which I can transfer the recombinant DNA into the specific host cell. Once it will get stably integrated into the host cell, it will produce its number of copies. So we will get the clone copies of this foreign gene, that is gene of insulin, into the bacterial cell. Now we are allowing that gene to express, that is that gene will get transcribed and then translated to produce the human insulin. Now the human insulin is inside the bacterial cell. I will cut that in the bacterial cell so that I will extract the human insulin. I will purify it, and then after purification, I can sell that insulin at the market as a human insulin. So this is a very nice technique by which we can use this cloning vector for our benefit. You can also transform such a you can prepare also certain plants to specific disease resistant or specific disease or specific insects by the same method. So there are different types of genetically modified organisms have prepared by the genetic engineers with the help of such a cloning vectors. For example, Monsanto tomatoes are there, then golden rice is there, etc. Now, if you want to select any DNA molecule as a cloning vector, it should have this following characteristic. One of the characteristic is it should have the self-replication mode, and it should synthesize its multiple copies. Whenever I will transfer that cloning vector into the specific host cell, it should not depend upon the host cell for the replication. It will replicate as itself and it will produce the multiple copies, as well as it must have the origin of replication, due to which it can self-produce. Also, it should have the cloning site at which we can transfer our gene. It should have the selectable marker gene by using which we can screen that host cell, in which host cell our recombinant vector is present and in which host cell it is absent. Obviously, it should be small in the size so that we can handle it in a very careful manner. It should have the low molecular weight. It can be easily isolated from the host cell as well, and it can be also purified in the easy manner. And as if we are expressing our gene into the specific host cell with the help of this cloning vector, so obviously we must have that cloning vector must have the region on the DNA which are useful for the transcription and translation purpose. That is, that cloning vector must have the promoter region, it should have the ribosome binding site, it must have the operator site, so that if I transform this recombinant vector into the host cell, it can express and it can produce our interested protein into the host cell in which we have transformed. Now this is a diagrammatic representation of the plasmid. Here it is a this is the cloning site which can be cut with the help of specific restriction enzyme, and which here we can attach our gene of interest. This is the origin of replication site, due to which it will not depend upon the host for the replication. It can produce its multiple copies, and it is having a selectable marker gene. With the help of this gene, I can identify uh, which bacterial cell have transformed easily and which bacterial cell has not shown any response to that cloning vector. Now, if we are want to define the plasmid, then it is a extra chromosomal. In the bacterial cell, apart from its chromosome, it consists of the circular DNA, which is having a self-replicating mode, which is a double-stranded circular one, and that is called as a plasmid. Its size vary from the bacteria to bacteria, approximately 1 to 200 kilobase pair. In certain bacteria, you will observe only single copy plasmid, but in several bacteria, you can identify at least 2 to 5 or more than that, that is multi-copy plasmid. And in the relaxed replication mode, obviously it can easily synthesize its 1,000 copies per cell. So this is the best choice for the used as a cloning vector. So, Cohen et al. For the first time reported this cloning DNA with the help of a plasmid as a cloning vector. And nowadays, whatever the natural plasmids are there, which are present in the bacterial cell, that have modified by the scientists, and they have prepared their own interested cloning vector, and which are referred as a artificial plasmids. So plasmid are suitable as a cloning vector because they are having a four best characteristic feature. As they can be easily isolated, they possess a restriction enzyme sites, they have independent mode of replication, and obviously they are easy to handle. These are the examples which are synthesized or which are prepared by the scientists in the laboratory. Here, all these are the artificial plasmids. Let us just see one of the artificial plasmid, that is PBr322. Now here, this small p is for the plasmid. This B, capital B and capital R is refers to the researchers who have discovered this plasmid or who have prepared this plasmid, that is Bolivar and Rodrigues. B for Bolivar, R for Rodrigues. And 322 number is numerical designation. That is, it is not like that just in the one step you are getting this artificial plasmid. They have to synthesize number of batches so that they can get the ideal cloning vector. So this is the 322 is the just numerical designation for this plasmid. Obviously, it is a high copy in the number when we will transfer this into the host cell. Approximately, you will get 20 to 30 copies per cell. It also consists of the two important antibiotic resistant gene, ampicillin and the tetracycline resistant gene. It also has the cleavage site for the restriction enzymes such as EcoR1, HIN3, BAM-H1, etc. And this cloning vector has designed by the scientists with the help of three different naturally occurring plasmid, that is R1, R65 and PMB1. Here, origin of replication is derived from the PMB1, and the unique it has a unique restriction sites for the 20 different restriction enzymes. So there is a multiple opportunity with this artificial cloning vector. Out of this 20 restriction enzymes site, it has a 6 within the tetracycline gene, 2 within the promoter region, and 3 within the beta lactamase gene, and due to which we can use the insertional inactivation technique so that we can identify the transform cell from the non-transform cell. So foreign gene can be cloned at any of these 11 sites of the restriction enzymes. So this is the restriction map. This is the map of PBR322, which is having a size of 4 to 4 kilobase pair, 4.4 kilobase pair, and these are the few restriction sites for uh, restriction enzymes. EcoR1, HEN3, SALH1. These are present in the tetracycline gene, and ampicillin, PST1 restriction enzyme site is present on the ampicillin resistant gene. This is the origin of a replication, which has synthesized. This is the artificial plasmid, which is synthesized by using the parts of the naturally occurring plasmid, and it is a very famous in the genetic engineering field because as it is having a number of a restriction enzyme site, and if I'm not having any of this restriction enzyme, I can use the alternative restriction enzymes to use this plasmid. I hope that you have understand this very nice plasmid which have synthesized by the genetic engineers. Thank you so much.